Hi, my name is Zach Marin. I want to thank the organizers of I2K for inviting me to talk to you about creating surface reconstructions of subcellular membranes, like the one shown in the background of this title slide, from single molecule localization microscopy or SMLM data. In SMLM, we are able to turn dye molecules on and off through time and localize their positions as they appear. This lets us build up a super resolved picture that gives us about 20 nanometer resolution of an underlying cellular structure. However, if we label a membrane protein shown here in green that's sitting on a membrane shown in blue with a dye shown in red, and we image it, then we're left with a bunch of dye locations and the uncertainty in those dye locations. And so this is great for improved resolution, but not great for looking at a membrane. So the question is, how do we go from a discontinuous pointillistic representation of the endoplasmic reticulum, such as what's shown on the left, to its surface representation using this kind of data? What people have done before is they've used a method called screen PYthon reconstruction, which is the state-of-the-art method for reconstructing surfaces from point clouds, specifically from LiDAR data and 3D range scanning data. And when it's applied to a very dense SMLM data set where all of the points have a low localization precision, then you can get a pretty good reconstruction. So up here on the left, I've densely labeled a theoretical 3D figure 8, and to the right, you see its corresponding screen PYthon reconstruction, colored by the distance from the reconstruction to the theoretical surface. So over here in red, you see that this part of the reconstruction is about 5 nanometers below the theoretical surface. Over here on the right in blue, you see that it's about 10 nanometers above the theoretical surface, and on average, it's about, according to this root mean square value, it's about 5 nanometers away from the theoretical surface, which is a value that's lower than the average localization precision for this data set, and so we say this reconstruction is pretty good. As the density of the point cloud decreases to more normal levels for SMLM datasets, screen PYthon reconstruction performs worse. It's not fully recovering the underlying structure. And if we further increase the localization precision or the noise or both to more reasonable values for an SMLM dataset, then SPR really has trouble fitting these points because it's trying to fit all of the points really well, and it ends up just sort of draping a sheet across all of them that tries to fit the background points as well as the actual structure points, and the result is something that looks nothing like the underlying structure. We realized why this is happening is because SPR is only using point locations, not the localization precision that is also available with the SMLM data. And so there's no existing technique that uses all available SMLM data when performing this kind of surface reconstruction. And so we built one. We start with the points and their localization precision. Initially, we just use the points to create a density-based isosurface where we cut a surface everywhere in the point cloud where we go from low density of points to a high density of points. And you can see that for the boxed region in white on the left, we get already a pretty good surface reconstruction shown here on the right that gives us a good idea of the shape of the membrane. In fact, everything in this reconstruction is a little bit of an over-approximation because when you cut a surface where the density goes from low to high, you're actually putting the surface a little bit outside of the actual locations of the points. This is published in our PYME Visualize open source software package, which is designed for interacting with, visualizing, and quantifying SMLM datasets. And you can look up the publication using the citation on the bottom of this slide. Once we have that initial surface, we perform hole and cut generation, sometimes depending on the region that gets used to estimate the density of the point cloud. You can choose a region that's a little too big and paper over empty regions of space. So for example, if we imagine this as a cutaway of two tubules running alongside each other into and out of the page, then we can see that we accidentally papered over the region in between the two tubules, but we can fix this by simply removing faces that can reach each other without passing through any points, and then patching the resulting holes. Once we've done that, we can remesh the surface to make sure that all the triangles on the surface are roughly equilateral, which is good for numerical stability for further operations. Then we can use the point attraction force to start bringing the surface towards the point locations subject to their localization precision. At the same time, we can look up bending stiffness coefficients for the lipid mixture of our membrane in the literature and feed this into a bending force, which helps make sure that the membrane remains smooth and doesn't bend in a way that an actual membrane couldn't bend while it's getting pulled towards these points. And then we balance the point attraction and the curvature force against one another and iteratively update the mesh position. And once the mesh positions are not, the mesh vertex positions are not updating very much from iteration to iteration, we say we've converged and we've found our membrane. In the event that after a few iterations convergence hasn't happened, we pass it back to the whole cut generation and remeshing steps just to make sure that we're still dealing with a high numerical quality mesh, and we go again. What this looks like with our toy example is I'll put the localization precisions back on so you can see what's happening. We smooth out our surface, we pull it towards the points, we smooth it out again, and you can see that the surface is allowed to float more near localizations with a high localization precision and has to adhere more strictly to points that have a lower localization precision. And if we do this, then we get a reconstruction that is almost as good as screen-Poisson reconstruction in the high density, low localization precision case. But as we decrease the density or increase the localization precision or the noise, our method continues to perform well where screen-Poisson reconstruction completely breaks down. So we have a way of reconstructing a surface in a more robust manner than SPR for SMLM data. We achieve a surface accuracy that is better than the localization precision of the data set. That is, our root mean square values are always better than the average localization precision. And we think that this should enable us to perform new types of biological studies. So we went ahead and applied this to real data. We fit a data set of Sec61 beta, which is an ER transmembrane protein marker. And you can see that zooming in, this fit is quite good. This came from 4Pi SMLM, which is a nice high resolution isotropic SMLM method. We also tried this on standard astigmatic SMLM data, and the fit is rather good for the quality of data we have, but we like to try it on more astigmatic data sets. If anyone has a good astigmatic data set and wants to share it with us, we'd be happy to give this a go, because this, in theory, should work equally well for any type of SMLM data. Just a quick note on measuring curvature in 3D for what we're going to talk about next. In 3D, there are two principal curvature directions. So for example, on a tube, one direction would be along the tube. You can imagine if you're walking forward and backwards along a tube, it appears very flat, and that's the minimal principal curvature direction. And then across the tubule is the maximal principal curvature direction, and this is 1 over the radius of the tubule for a perfect tubule. So if we plot the principal curvature, the maximal principal curvature, the curvature across the tubule for the data set that these ER images arise from, there's actually a lot more ER in this data set than I'm showing here, but you can see that it includes some ER sheets. So if we look at the median maximal principal curvature for this data set, we see that it's 1 over 53 nanometers, which means that our average tubule diameter is somewhere around 106 nanometers in this data set, which is smack dab in the middle of the range that we would expect. So this is good validation that this method is working on real SMLM data. My lab mate, Lukas Fuentes, took this beautiful ER data set in lamellipodia, and we can reconstruct the whole thing. And he also, at the same time, imaged ER structural proteins called reticulin 4, which are thought to give the ER its highly tubular structure. So basically, they're found to localize in areas that are high in curvature on the ER. So they wrap themselves around tubules and localize to the sides of ER sheets. And in fact, that is exactly what we see when we plot them on top of our ER surface reconstruction here. We can actually quantify this, so we can again look at the maximal principal curvature direction on this ER data set, but we can also look at the density of reticulons near vertices of each of these curvatures, and we can see that the reticulon 4 density is actually a bit higher in ER tubule, or regions of the ER that have a slightly lower or slightly higher curvature, or a narrower tubule diameter. And if we plot the ratio of these two histograms against one another, we actually can see that as we move into areas of this surface reconstruction with higher maximal principal curvatures, the density of reticulons in those areas also increases all the way up to about 1/16 nanometers, or indicating that the smallest ER tubule in this whole data set is something like 32 nanometers in diameter, which is again exactly what we would expect for this kind of data set. And of course, we would also expect this pattern of reticulons increasing in density in areas of higher curvature. So again, I think this is excellent validation that we're really getting quantitative surfaces out of this reconstruction technique. I'll just show you quickly the ER with some mitochondria as well. You can see that the mitochondria here are just snaking through the little holes in the ER, which is quite cool, and you could use some sort of reconstruction of these two channels together like this to look at ER mitochondrial contacts. We've also meshed transmedial and cis Golgi, and just to show that this method also works with non-membranous structures collected in SMLM that are closed, I also meshed some tubules, microtubules, just so you can see that that works as well, even if it's not a membrane. With that, I'd just like to say, yeah, you can download this at pythonmicroscopy.org. The initial surface reconstruction is already there. The iterative fitting described in this talk is still in a private repo, but if you're keen to try it right away, just get in contact with me. I'd like to say many thanks to my advisors, Joerg Bewersdorf and David Baddeley. David actually wrote the software with me, and then a big thanks to my lab mate, Lukas Fuentes, who did all of the biology, and it's just been awesome to work with. And with that, I will take any questions.